So can we have the next slide, please?
Hopefully.
Yeah, thank you. So just a quick overview of the topics that we'll be kind of covering today. So basically the overarching theme of the talk is the physiological responses to nutrients in humans. So we'll kind of talk about gut effects and signaling, also the gut-brain axis, and also the impact on appetite, and also roles in health and disease. But one of the major kind of focuses of this talk are kind of the future directions and kind of the undiscovered things, the things that haven't really been looked into that much, because there's a lot more in this topic than you think that hasn't actually been researched yet. So it's quite an exciting and diverse kind of upcoming field. So yeah, should hopefully be a good talk. That's what we'll be covering today. Yeah, so just kind of like to start at the very basics, I guess. So gastrointestinal anatomy and physiology were adapted to absorb ingested nutrients with maximal efficiency, because when you think about it, kind of if you go back to when, you know, evolution started, and, you know, prehistorically, you were never really sure when your next meal was going to be coming. And it kind of wasn't something where food is as readily available as it was now. So kind of we were programmed innately to kind of have a focus on survival. So there's nothing actually wrong with our gut biome. It's just kind of the fact that our environment has changed and not our biology. So yeah, kind of just summarizing this little yellow kind of bit. Eating, digestion and absorption are essential, obviously, and they're also highly evolved and closely regulated. But I mean, the kind of key mechanism that humans still have today is eat as much as you can when it's available, because you don't know when you're going to have your next meal. And as we kind of said, even though that's not as applicable now, our programming in the GI system is still kind of to do with that, as it would have been in prehistoric times. So crops, cooking and processing are recent and only human developments. So our biology is outdated, but fixed at the same time. So yeah. And those are just some pictures to kind of illustrate that. So
yeah, it's not just a human thing.
Mm-hmm. Okay, so kind of just going into, I guess, the cells themselves a little bit. So the gut epithelium is the largest endocrine and sensory organ in the body, which is quite surprising because no one ever thinks of it as being kind of, you know, that big or that relevant. And then in this picture, so it's an electron micrograph picture of gut endocrine cells. And so where the arrow is, that's the apical microvilli. So they express chemosensory receptors. Is there more text to come on the side? Yeah, okay. So yeah, with the chemosensory receptors, they kind of help us with distinguishing different tastes like sweet and bitter ones, and also fatty acid receptors, and also immunological receptors, which once again is quite interesting because I guess we don't always associate gut epithelium with really being that important in the immune response, but it is. So that's kind of just an overview. And then this little diagram kind of summarizes quite nicely how there's paracrine and endocrine signaling going on. So I guess people are mostly familiar with endocrine signaling with, you know, hormones like thyroid hormones and insulin, where kind of effects occur from a distance. But then there's also paracrine signaling where you've got your effects happening a lot more immediately adjacent to cells. So the diagram kind of shows that you've got this variety, as John is very kindly showing with his cursor, and you've got a variety in kind of where you're going to have your effects. And you can also see the apical and basolateral kind of surfaces of the cells in comparison to where they are relative to everything else. And yeah, essentially enteroendocrine cells sense nutrients, and that's why they're so key to how we exist today. Okay, so to kind of move on to this picture. So basically EEC, I'm just going to abbreviate them because it's a lot easier to say. They release gut hormones in response to the luminal environment. And because they're scattered throughout the intestinal epithelium, they're kind of widespread, but they're also quite rare. So they've got long microvilli that kind of taste luminal content. And there are also lots of different subtypes, which is obviously good because the more subtypes you have, the more kind of diversity you have in the range of functions they have, and something that we've kind of adapted to have. And just to talk a little bit about the picture, it's immunochemistry picture, and so quite interestingly, actually, I'll go through the hormone, and then I'll say my fun little fact. So I won't spend too much time going through all of the gut hormones, but I'll do a little bit just to look at everything. So we've got gastrin, and basically amino acids are the stimuli for gastrin, secreted by G cells. And then secretin stimulate fat and pH, and they're secreted by S cells. And this was my fun little fact. Secretin was actually the first hormone that was ever discovered by Bayliss and Starling. Everyone kind of always assumed it's insulin, but it wasn't. So if you've learnt one thing from today, hopefully it's that. So that can be a fun new little fact. And then you've also got CCK, secreted by I cells, which fat and protein are the stimulators for that. And then you've also got GIP, which is secreted by K cells, and they stimulate a kind of glucose and fat. So you're probably noticing a pattern that fat is a stimulant of pretty much all the gut hormones, which is what you'd expect. And you've also got PYY and GLP-1, which is secreted by L cells, and fat and carbohydrate stimulate those. And fat and carbohydrate also stimulate tensin, which is secreted by N cells. So yeah, I won't go into more detail about that, but that's kind of like a nice little overview just to contextualize everything. Yeah, so once again, this kind of slide just shows that there's lots of different receptors and that it's quite a complex overarching system, but it allows us to kind of have really specific and really diverse reactions to our environments, basically. So, I mean, I won't go through all of the different kind of receptors, but as you can see, there's lots of different ones. So they vary in their kind of chain length and also their GPRs. And they also help us taste. So yeah, I don't know if John, you want to add anything to that? No? Okay. Yeah. So I mean, once again, this diagram kind of just shows that there's a massive kind of neurological component to our GI system, which once again is a connection that kind of gets overlooked in terms of the brain-gut axis. So I mean, in terms of regulatory peptides, you've got stuff like CCK and GLP-1 and 2 and PYY. But then it's kind of thinking about how, in terms of the vagus system, these actually interact with the brainstem and the hypothalamus. So you've got your vagal afferents and efferents that kind of form the link between the two. And then obviously you've got nutrients in your actual GI system. And I guess one of the most important and I guess interesting links between this is genetic obesity. Because if you've kind of got issues in your hypothalamus, then that kind of affects your gene expression. And that is something that kind of affects genetic obesity, which is rare, but it's a common misconception that all obesity is caused by kind of lifestyle changes. And no one really kind of ever thinks about the fact that some obesity is in fact inherited. So that's quite interesting, because essentially in syndromes like Prader-Willi syndrome, you don't get signals to stop eating, so you just continue eating no matter whether you're full or not. And that's, yeah, something that I thought was quite interesting.
Leave that.
Yeah. Okay, so just a little bit about vagal receptors. I think this diagram is quite nice because it seems a bit busy, but it kind of illustrates the main points really nicely. So you've got receptors being expressed in nodose ganglion cell bodies, and then your afferent nerve fiber terminals are going to display receptors. So it's kind of unclear as to what the full receptor complement actually is. But you've also got chemical, hormonal, mechanical, and physical and immune sensors all being present. So that really kind of contextualizes and puts into context just how important this system is and why it's so crucial that it firstly functions properly, but also what kind of pathologies can arise if it doesn't, because it encompasses basically all the different constituents that we need to function normally every day.
It's not going down.
Okay, awesome. So yeah, this diagram is kind of something that really summarizes, I guess, one of the biggest kind of components in the GI system, which is what inhibits food intake and what stimulates food intake, because obviously having a balance of them and having things that work when they should work is once again kind of key to having a normal daily life. So in terms of the stomach, you've got leptin inhibiting food intake, then you've got ghrelin and orexin stimulating it. So, you know, one of the things you kind of learn in like GCSE biology is ghrelin makes you hungry. And that's kind of where that comes from. And so it's also important, I guess, to note that it's kind of very compartmentalized as to where you'll find these different stimulants. For example, you know, you'll find these mainly in the stomach, whereas if you go to the upper small intestine, you'll find CCK, which is the main inhibitor of food intake. And then you've got anandamide, which is going to stimulate it. And anandamide is not actually a hormone, it's linked to the cannabinoid system, but that's just, once again, something that you'll find in the upper small intestine. And then in the ileum and the colon, you've got GLP-1 and PYY, which are both going to inhibit food intake. So you can kind of see that it's a system of checks and balances. And when they all work together in harmony, you've got a kind of regular, normal intake of food that's going to be going on. But evidence for the physiological effects on food intake in free living healthy humans is scarce. Most of the kind of research is based on animals, but obviously there's a big difference between kind of digestion and the GI system in animals compared to humans. But it's quite hard to kind of do research on this topic. So our kind of knowledge is limited, but we know basics like this, which can help us in the future kind of discover even more. Yeah, that's just a nice little picture to brighten up your Wednesday evening on this cold, wet evening.
Yeah, thanks, Kiran. That was very clear. Nice presentation in the background. I guess this is part of the problem, in a sense, just a little bit of a joke, but we know that given our programming to overeat, that's what will happen potentially. Maybe not quite to that extent. That would be quite excessive, but. Right, so I'm going to go to some of our Manchester research over the years. This is going back quite a way. Actually, this is actually the main finding of my PhD, which I did in Manchester. But it's really a lot of the future research is based on this. So as Kiran said, that fatty acids are one of the things that release the hormone CCK, and CCK is the hormone that when you eat fat, it stimulates the gallbladder to contract, amongst other things, which is how it got its name. But we didn't really know what fatty acids produced CCK. So this is experiments we did in humans with healthy volunteers, including myself. Actually, I was a volunteer for my own studies, so that putting nasogastric tubes down myself and cannulating myself to take blood samples, and we were infusing these fatty acids into the gut to see if they have different effects. Sorry, it's gone. So different fatty acids have got different chain lengths. So there's 4 and 10 are short chain fatty acids, and medium chain fatty acids, and then 12 and 18 are longer chain fatty acids. But the interesting thing was just there's this very big difference in CCK release just with a single carbon atom change. So going from 11 to 11 carbon atoms in the fatty acid produce pretty much no response, and this tiny response to 12 induces this full meal-sized response, which didn't make immediate sense. But then we discovered if you look at the solubility of the fatty acids and the temperature at which they melt, there's a big difference between C11 and C12. And C12 is the first fatty acid that really needs bile to dissolve it. So the system, and as I said, CCK induces bile secretion. So the first fatty acids that's sent to make the gallbladder contract is the first one that requires bile. So really quite a sort of a sophisticated thing within the system. And then we went on to look at whether this had differential effects on various aspects of physiology, which I won't go into today. We also got very much into the cell biology of fatty acid sensing. So we know that endocrine cells, when they have a stimulus, it's mobilization of intracellular calcium that causes the exocytosis of the hormone or the cellular products. And so we wanted to know if we'd seen this in whole humans, but we wanted to see if this was true in the endocrine cells themselves. So there's a cell line that we can use in the laboratory that's a CCK secreting endocrine cell, and so we tried the fatty acids on this, and remarkably we had exactly the same thing. So our C8 and C10 fatty acids didn't really cause an increase in calcium. The y-axis on this is the calcium level inside the cell. But when you when Fura2 is a dye that fluoresces in response to calcium. But when you increase the fatty acid to the C12 that have the effect in the humans, you got this huge spike in calcium in the cell and release of CCK. So that means whatever the mechanism that's responsible for this in intact humans is clearly present in the cell itself. It's right down to the individual cell level. So the problem is, I think, as Kiran alluded to before, that it's difficult to study the gut endocrine system because that. So see, so if you want to look at the pituitary, it's relatively easy because you can access the pituitary, remove the pituitary and study it. Or you can do the same with the thyroid or even the islets in the pancreas. But because our endocrine cells are single cells scattered through the epithelium, it's really difficult to study them in situ. So we're hoping to be able to move towards that by this methodology, and because it's not possible to kind of isolate the cells. But what we've been able to do is put use the CCK promoter. So that's the thing that controls the CCK gene to be switched on in cells. But you attach that then not to CCK but to another fluorescent protein. So this thing called a green fluorescent protein, which comes from jellyfish. And the good news, it worked. So we know that that's our endocrine cell from the micrograph before. But when we've used the CCK gene to put this fluorescent protein into the cells, then we get this nice staining of just the CCK cells. And people have done this with other gut hormones as well. So the hope is now we'll be able to do things to isolate those cells and be able to study them in the laboratory. And the other good thing, when we have isolated those cells, then we found that this is PCR looking at the fatty acid sensing receptors. So GPR40 and GPR120, which Kiran mentioned earlier, the fatty acid centers are both present in the cells that we've sorted using GFP. So these are the CCK secreting cells, but in the other cells that aren't CCK secreting cells, the receptors are absent. So it looks pretty much like we've nailed it down to the fact that the endocrine cells themselves are the fatty acid sensors, as the model suggested, but also that these specific new fatty acid receptors are in those cells. So those are potential targets for the physiological role is to respond to fatty acids, but of course they're potential drug targets as well. So in theory, you could produce things that stimulated these pathways, and so the gut would think that there was food in it, but without actually being food in it. So moving up to the brain, so we can do these things in the gut. The problem is that there's a limit to what you can do in whole humans in the gut, because probably these hormones are present in the portal circulation, and then they're rapidly metabolized in the liver. So there's a limit to what you can measure in peripheral blood. But what we can do is look at the central nervous system correlates of this using this technique of functional MRI, which you might be familiar with. But in a nutshell, it looks at the ratio of oxygenated to deoxygenated hemoglobin, because the oxygenated hemoglobin has got a slightly different magnetic signal, so behaves a little differently in an MR scanner. And when you're using part of the brain, that part of the brain takes up more oxygen, and therefore you get a slight increase in deoxyhemoglobin. And using sophisticated MR techniques, which I don't remotely understand, you can use this to map things onto the brain. So you can, and what's called the BOLD signal, the blood oxygen level dependent signal, and you can map this as kind of increases or decreases in BOLD signal to map what's happening in the brain. So there's been lots of this done with kind of movement physiology and listening, visual physiology and auditory physiology, and in different health states, but it hadn't really been done very much from the gut, and it wasn't entirely clear how possible it would be. But it turns out it was very possible. And so when we put our C12 fatty acid, the one that causes the CCK secretion, into the gut, then all these areas of the brain are activated so nicely. The hypothalamus and the brainstem, where we know that the vagus nerve terminates, were activated quite nicely. But then you also start to see kind of higher centers of the brain being activated, and these are probably things that have got some relationship to appetite control and food ingestion. The thing we've never understood is why fatty acids in the gut induce some activity in the cerebellum. So the one slightly amusing idea is maybe that's the whole meaning of a balanced diet, but I don't think that's very likely. And then the other thing we can do with CCK, there's a drug called dexloxyglucomide, or Dex, on this graph, and it turns, and that blocks the CCK receptor. So in the red line here, that's the time course of brainstem activation in response to the fatty acid in the gut if we give a placebo for the CCK receptor. If we give saline, not fatty acid, nothing really happens. But if we give the dexloxyglucomide antagonist, that's the yellow line, to block the effect of CCK on the receptors, then this rise of brainstem activity doesn't happen. So we think what's proper, I mean, and we know that the CCK receptors are expressed on the vagus nerve afferents that Kiran illustrated. So we're pretty clear in this, we're blocking the activation of the vagus nerve and therefore preventing the signaling to the brain. I'm not going to go through this slide. This is just to kind of point out that there's a whole complicated brain chemistry. We've got all of this stuff that's happening in the gut and all the gut endocrine system, but there's a big neuroendocrinology set of signals and receptors in the brain as well. So it's a very complicated system, but our focus is on the gut end of it. Another interesting thing, I think this is something we're taking on board for future studies. It turns out appetite has got quite a marked circadian rhythm. So if you look at, without feeding people, if you look at how hungry they are on a visual score over the day, you get this kind of quite marked changes, so that people tend to be less hungry in the morning, even if they're not fed, and more hungry in the evening, and with this cycle that goes on. So one thing we're starting to think about is when we're looking at our experiments, we tend to always do them fasted first thing in the morning, because that's kind of the convenient time to do experiments. But of course, if we're trying to look at things that control hunger and appetite, it might be better for us to be doing the experiments at 8 o'clock in the evening, because that's when people are the most hungry. However, getting people to fast all day and come in for an 8 o'clock experiment in the evening and having the researchers to do 8 o'clock in the evening experiments is obviously a little bit more challenging, but something that we need to take on board. But of course, as Kiran said, the hedonic drive, so hedonism, that's the pleasure that you get out of feeding. So there's no doubt that eating food is a satisfying thing. Unless except in illness, it's a nice sensation, and it's a normal thing to do. So a lot of this is therefore about the biology and what controls things from a gut to brain signal. But of course, there's a whole cognitive thing going on that people choose to eat, and we know that if there's nice food that people like, they'll continue to eat it even though they're full physiologically. So there's this tension between the liking and wanting of food and what we might call the neurocognitive responses, and then the more biological basic things of the food microenvironment in the gut and metabolic status. So that's just to illustrate what a complicated situation and setup it is, and we can't just look at the gut in isolation or the brain in isolation. Both of these things in humans are competing. I mean, and this is true in rodent experiments, for example. You know, if normal rodent food in a cage is just dry pellets, which I'm sure are not very interesting, but if you put sugar or fat in rat food, then the rats will preferentially go for the sweet food or the fatty food. So that kind of drive to have the more pleasurable, satisfying foods is really preserved right down to other animal models. And again, this is just to illustrate the kind of pathways between the different cues and signals that are all interacting in how we eat. And of course, then human beings are very different. If some human beings will be very much weighted towards the overeating end of the spectrum, and others very much towards the rapidly satisfied end of the spectrum. And then you've got to put that in the context of the environment that people are in, so it all becomes very complicated. So this is one thing we just done recently. We've developed this package of a computer-based model where we can look at people's kind of what we call an attentional bias score. So this is when you show pictures of food, you can measure how quickly people click on the pictures of food, and then you use other images, and we use actually envelopes and pens and things as non-food images, and people don't click on those very quickly at all. And when you can work out this thing called the attentional bias score. So when people are fasted, they go for the food images quite quickly and click, and there's a significant. But when they've been fed, then that switches off. So it's quite a nice model potentially. So we can start to explore the kind of the relationship between different types of food in different disease situations as well, using these attentional bias scores. I really didn't think that was going to work when we first did it. It originally came from alcohol and drug related research. So people with alcohol and drug dependencies click really quickly on the alcohol or the drug images, and I really didn't think it worked just for food, but it did. So there's a lot of ideas whether these things actually regulate food intake, the physiological pathways, but most of the things, as Kiran said, are from animal studies. But if you look at the things that have been done in humans, they're almost always where there's been an infusion of the peptide. So you infuse GLP-1 or infuse CCK or infuse PYY or whatever. And then you get really very high levels when you infuse it intravenously. You get very high levels of those hormones, like 3.64-fold or 3.1-fold before you start to suppress energy intake. But if you look at actual feeding studies, you only get that higher CCK level in about 29% of studies. No study has ever generated a 4-fold increase in GLP-1 induced by eating, and only 8% of studies have shown a 3.1-fold increase in PYY. So there's a question there whether actually using food to release these peptides endogenously really can be very effective as a satiety pathway, and this maybe have to be more of a pharmacological approach than a nutritional one. So it just leads to this question about whether gut hormones actually regulate food intake, and although there's no doubt there's a physiological role, I think we're just increasingly unclear whether these gut hormones regulate normal food intake. And back to the evolutionary thing, why would you? It wouldn't make sense to have a sort of a system that when as soon as you've got lots of nice food that you've been waiting for days, that as soon as you're slightly full, you stop eating. That just wouldn't be the correct evolutionary drive. And that's just another thing to show that that endogenous GLP-1 is a weak satiety signal, because if you infuse a GLP-1 receptor antagonist, it really makes very little difference to the desire to eat. So it's probably endogenous signals have a limited effect. So could potent nutrients or drugs acting on gut nutrient or hormone receptors be clinically useful in reducing appetite or weight? Well, when I started my PhD, I really liked, 20 odd years ago, that's what I really like to think, and this will be a fantastic way forward for the future. But I think it's unlikely that the signals are really potent enough to do that. The evidence is weak, and these long-acting agonists in supraphysiological high doses may be effective, and there's growing evidence for that. And in fact, GLP-1 injections are already in clinical practice. That slide's not showing very well on my screen, so I'll skip it. Okay, so let's be turning more clinical. So we know about kind of this maybe being not so important in health, but what about enteroendocrine cells in disease? So of course the difference then is not so much if people have got gastrointestinal disease, it's not a tendency to overeat, it's a tendency to be not hungry, and we really don't know what the underlying mechanisms for that are. Then this is another thing for my PhD. So my PhD supervisor was one of my healthy volunteers for one of my meal studies, and this was when we were using chicken soup as a test meal to measure CCK responses to chicken soup. And as I mentioned before, in healthy controls, you get this really small, doubling of CCK, not in response to that. But he took part in one of my studies, and when I measured his CCK levels on the Friday afternoon, they came back about 50 times above the normal range, which was very strange. And I thought he must have an endocrine tumor in his gut. So I spent the weekend worrying about how I was going to tell my supervisor on Monday that he probably needs to go for a scan, because he's looks like he's got a gut endocrine secreting hormone. And he said, "Oh, I'm really sorry, I've been really ill over the weekend. I'd just come back from an overseas trip, and it turned out he got this parasite Giardia, and so he shouldn't have taken part in my experiments as a healthy volunteer, and should have told me that he was poorly. But it's quite a nice discovery. So we managed to do this in about I think about 10 more people with Giardia, and you got this persistent elevated rise in CCK at baseline, and this really big rise in CCK levels in response to the chicken soup, much bigger than normally. And the interesting thing about Giardia is it lives in the upper small intestine, so it lives exactly where the CCK cells are. And when these patients had the Giardia eradicated, a couple of them let us test them again, and the levels had returned to normal. So this is really the first potential evidence that gut hormone responses were altered in gastrointestinal infection. So that might suggest that although we've got these normal pathways in physiologically, that they're perhaps in disease. So if you've got an inflamed gut, there's going to be an amplification of these gut peptide derived gut brain vagal signaling pathways. So that's what we think is probably going on. If you remember the STC1 cells that we used for the physiology experiments, so we went back to those and thought, let's give them a cocktail of the cytokines that you see in Giardia infection. And if we incubated the STC1 cells with pro-inflammatory cytokines, then we got this big increase in CCK release from the gut cell line. So again, it seems that the cells themselves, as well as sensing the fatty acids, the cells are sensing the inflammatory response and responding with an increase in CCK secretion. So probably that's if these cells have got the fat receptors and they've got the immune receptors, there's probably a biological rationale for that, so that to have increased CCK responses in response to a gut infection is probably an adapted physiological response. And it may be that actually switching off appetite and stopping eating and drinking is then a kind of a programmed response that's appropriate if you need to rest your gut and not take any more food in, then to have an alarm system that switches it off and does it very quickly. Within 2 hours, we were seeing that. So I think this is probably actually a function of the endocrine cells. So we started wondering about how the inflammation affects CCK in vivo. So there's lots of experimental models for causing intestinal inflammation, chemical or biological ones. But because of strengths in the immunology department in Manchester, we chose to use this nematode worm called Trichinella spiralis, which is a topic of research in the university already. And obviously that's a very magnified picture. They're very, very tiny, really. But so Trichinella spiralis is what's historically you get from raw or undercooked pork, which is why we avoid eating undercooked pig meat, or for anybody, because it causes this transient problem. It goes back to normal. So clearly, so this was looking like a good model for understanding how infection was affecting these pathways. And if we give the loxyglumide, the antagonist we'd used in the human brain experiments, then that also reversed the reversed. So in food intake in uninfected mice reduced, but then if we give the CCK antagonist, they eat almost back to normal. And the strongest piece of evidence was then we got hold of some CCK knockout mice. So the gene for CCK has been deleted in these mice. They don't have it at all. And when these mice are infected, when you'd normally hear you'd start to see this drop in food intake, it doesn't happen. And if anything, they actually start to eat a bit more, although that wasn't significant. So I think we felt fairly confident now that CCK is the thing that's mediating reduced food intake in acute gut inflammation. And interestingly, obviously, the CCK knockout mice, you'd expect them to be overweight if CCK was very important in food intake, but they're not. And then we went on to look at the immune mechanisms for this using these mice called SCID mice. So they don't have T or B cells. And again, if you infect these mice, they don't have the drop in food intake that you'd expect. And therefore that suggests that it's something in the immune system that's driving this. And it turned out to be CD4 T cells, because if we gave CD4 T cells from infected normal immune system mice and put that into the immune deficient mice, then they did have the drop in food intake that you'd expect. So now we've got this quite clear axis between CCK, the vagus CCK receptors, and CD4 T cells being the thing that drives the change in endocrine cell function. And again, I've suggested before, maybe this is playing a purpose, because in the CCK knockout mice, then it actually takes them longer to clear the worms. So at 15 days, most of the healthy mice have got rid of the worms from the gut. whereas the CCK knockout mice have still got twice as many worms. So we think that rather than just being an appetite regulating thing, this is probably part of the response to the infection. So the CCK cells are playing a direct response in the pathogenic mechanisms in response to infection. And this one was quite nice. This got picked up by BBC News, which was nice in one way, except they got it completely the wrong way around and decided that this was proving that weight loss was the body's way of fighting off gut worms, which of course wasn't what we thought at all. But always got to be careful what you read. So we want to start to think about human gut inflammation. So the easiest way to do that is with ulcerative colitis, because you can do rectal biopsies quite easily. And the rectum has endocrine cells. But curiously, when we looked in colitis in humans, there was actually kind of quite significant reduction in endocrine cells in the rectum, which is a bit disappointing. But of course, the rectum isn't actually involved in nutrient sensing. All that happens in the proximal gut. So we knew from our giardia that the duodenum and CCK cells were being changed by giardia infection. Whereas the other big area of nutrient sensing is the terminal ileum, where the GLP-1 and PYY cells are. And of course, that's the most common place for Crohn's disease to affect. Harder to get tissue samples of clearly, but not impossible. So Crohn's disease became our next focus. And so just some summary of this was that we know that people with Crohn's disease, even when they're fasting, feel more full, feel more distended, they feel more bloated. So that's probably all part of the, and in response to a meal, that Crohn's disease patients have much bigger kind of symptoms from the gut than healthy controls. And then we looked at hormones. So I mentioned the GLP-1 and PYY being the hormones expressed in the terminal ileum, and both of these fasting and across a time course after a meal was significantly elevated in the Crohn's disease patients compared with the healthy volunteers. So again, these are kind of far bigger increases in gut hormone levels than you'd ever induce with a meal. And we think this is part of the pathophysiological response. Of course, just as an aside, we've always got to think in humans, there's just so many things that can affect food intake and malnutrition. So I'm not suggesting at all that gut hormones are going to be the sole answer to this. But of course, if people are feeling sick or vomiting or get pain when they eat, then there's also all these factors are going to potentially interfere with food intake. So it's just to remind ourselves that it's a very complicated system. So we're back to you now, Kiran.
Yeah. So thanks for that, John. I thought that was really interesting. So yeah, just to kind of conclude, so nutrients induce gut peptide hormone signaling. So they regulate not only digestion, but also absorption and motility. And I feel like digestion and absorption are things that people classically associate with gut peptide hormone signaling, but always forget about the motility of it. But obviously all three are essential. It's kind of keeping us alive, really. So they're biologically appropriate and also efficient. But it's unclear if these really reduce appetite or food consumption physiologically. As John kind of mentioned, there's still quite a lot of uncertainty in the research field, but they might contribute to responses to gut injury and inflammation. Once again, that's kind of something where there's a lot more scope to kind of go a bit deeper in terms of research. And then, yeah, in terms of human disease, there's a big gap in kind of evidence and what's actually known. But for example, Crohn's disease, increased EE cells and peptide levels are kind of factors that might play a big role. But there's a lot more kind of work that can be done there. And then similarly, in terms of vagal damage with diabetic gastroenteropathy, quite hard to say. And when we kind of discuss the kind of role that the vagal system has in terms of the GI system, that's kind of a key area for research, really, but once again, something that's not really been touched on. But obesity, aging, and then also cancer are where roles of gut hormones and gut to brain signaling really need research. And it's quite, like we said, an exciting and evolving field. And there's plenty more kind of questions that need to be answered. And it's kind of surprising that these three kind of key areas have been overlooked. But I guess it's something where in the future, you know, there are potential for real groundbreaking kind of new discoveries can be made. Yeah, so just to kind of, you know, finish off with some challenges or potential products. So do gut to brain signals have a real life impact on the control of appetite and food intake or not? And also whether top-down hedonic drives simply override the physiological responses, especially now in a world which is kind of, you know, facing a massive obesity crisis. And also kind of looking at what the GI signals could be manipulated to either increase or reduce food intake, depending on what your desired outcome was. And also just generally how we can better study gastrointestinal sensing and signaling in humans, because as we kind of said, it's something that's only significantly been done in animals. So yeah. Yeah, and also whether abnormal signaling plays a role in obesity or in GI disease, because if, you know, you can kind of get to the bottom of what's going wrong in a signaling cascade, you can basically affect everything else going on downstream and play a massive impact in kind of sorting any disease. Yeah, and also kind of just looking at whether there are disorders of the enteroendocrine system itself, because there's roughly 20 different cell types and there's multiple hormones. So when all of those kind of interact and come together, there's massive scope for kind of pathology to occur. And all other endocrine organs have hypo and hypersecretory disorders. For example, you know, if you look at the thyroid hormones or kind of like others like that, we know a lot about those conditions and they're something that are quite regularly studied. But in terms of like endocrine organs relating to what we've kind of discussed, there's not really much out there. So there might be undiscovered disorders of EEC, and, you know, having to also consider what they would look like clinically is relevant, because if suddenly you get an influx of patients presenting with symptoms of this, unless you really know the kind of background history of what you'd be looking for, it'd be really hard to diagnose these people and ultimately.
I've not come across anything about oxytocin. Is that something you've seen information about somewhere else? I know obviously there's lots of brain peptides and hypothalamic peptides are involved in appetite regulation, but I must confess I've not seen oxytocin, but because I mainly focus on the gut. Oh, your dissertation's on it. Oh, that's good. Right, you have to send me a copy. I'll send you. So, yeah, that which dissertation was that from? And then, yeah, I'd be interested to hear about that if you want to send me some information or a copy.
And as Hamna also put in the chat, there's a feedback form. So once that's completed, you also get a certificate of kind of attendance of being here tonight. So the link is in the chat.
Brilliant. So if there's no more questions, I guess we should conclude. That's a good question. It's a little bit of a how Samarth asked, how do you specialize in enteroendocrinology? So you can't really, doesn't really exist as a specialty at the moment. I think it's comes within gastroenterology. I was quite after medical school, I wanted to do medicine. I wasn't sure if I wanted to do endocrinology or gastroenterology, and I decided, because I wanted to do more practical things like endoscopy, I went to the gastroenterology side of things, but got interested in gut hormones as a sort of a physiology thing at the moment, because the only things that are really recognized in clinical practice are endocrine tumors, so neuroendocrine tumors in the gut, and they're generally looked after by oncologists. But I think this idea that there might be disorders of the enteroendocrine cells themselves is still very much just a research thing at the moment. It's not come into clinical practice. So I think it's, as we identify more and more potential pathology, I think that'll come. But at the moment, it's really just at the research stage, apart from those endocrine tumours. Hamda's asked, how does bariatric treatment affect gut to brain signaling? Well, there's quite strong evidence that the gut hormones may be involved in bariatric surgery. So in particular, GLP-1 and PYY, that are the two hormones that are upregulated in Crohn's, are very much upregulated after after a meal in bariatric surgery, and that's probably because the proximal gut has been taken out of circuit. In most bariatric surgery, the nutrients get much further down the gut than they normally would. So normally the terminal ileum doesn't see much nutrient because it's all been absorbed before it gets there. But so in bariatric surgery, then because nutrients get to the place where they're not supposed to go, then they do cause a big increase in GLP-1 and PYY release. And people have done experiments where they've given antagonist infusions to GLP-1 and PYY in post-bariatric patients, and they get very hungry and they want to eat more. And things, an anecdote about one of the subjects in the study said, please never do that to me again, because they were quite terrified by the fact that they suddenly wanted to eat again in a way that they hadn't done before the bariatric treatment. There's some possibility that there may be sort of things that happen to the vagus nerve. So if the vagus nerves get sectioned, then that might have some part of it. I'm not aware of any brain imaging post-bariatric surgery, so I don't know if that's that top end of the signalling. I don't know if that's been done, but there is good evidence that gut hormones may well be involved in the response. And again, but that's probably why injecting large doses of those hormones as drugs also has an effect. It's just not as dramatic as bariatric surgery. Thanks Max for offering me a copy. I'll be interested. Does being a gastroenterologist research you do? Yeah, I think it's difficult. So I think one of the problems of gut researchers in humans is it's difficult to access a lot of the thing. There's a limit to what you can do. So you can put tubes in the top and the bottom end and look at things and measure simple responses, but you can't really sample what you're most interested in, because the gut hormones are mainly working in the paracrine system and mainly working and then rapidly metabolized in the liver. So it is restricted, and that's why we've got to go into these animal models to really understand things. But I think you just got to. There's a limit to the other thing with the gut is very variable in response. I think things like respiratory physiology and cardiac physiology in health are quite conserved and very similar between people. But the gut, you can have, some people might have a transit from the mouth to the colon in 20 minutes, and that's normal for them. But in somebody else, it might be 2 hours, and that's normal for them. So it's really so it's one of the difficulties with gastroenterology research is because it's got such huge physiological variability, and it's quite inaccessible. But I guess I'd like to see that as a challenge rather than as a that is a typo. Yeah. So I think it's an interesting thing. But I've actually stopped that. I mean, for me, it's now it's more that I do less gastroenterology. I only do 2 outpatient clinics a week now, and I've stopped doing endoscopy because I focus more on the research and the university roles that I have. But it's fun. Okay, I think the questions have stopped coming in. Oh, thank you. So thanks everybody for giving up an hour of your evening as well. It's nice to nice to have a chance to do this, and wish you all the best for Christmas and the New Year.
Yep. Thank you very much to our speakers this evening, Professor McLaughlin and Kiran. Yeah, it was super interesting. I learned all about the new research, and special thanks to Professor McLaughlin for being our supervisor for this talk series. It's been absolutely lovely to have you. And thank you to the participants as well. I hope you found this talk really useful, and I'd just like to remind you that if you could just fill in the feedback form that we'll be sending out afterwards, it really won't take you that very long. And yeah, have a wonderful evening. Thank you very much for coming.
Thank you.